During interphase, a cell that is about to divide grows and copies its chromosomes in preparation for cell division. Interphase can be divided into subphases. The G1 phase stands for first gap, the S phase stands for synthesis, and the G2 phase, which is the second gap. During the G1 phase, the cell increases in the mass and size for preparation of cell division. The S phase, the period during which DNA is synthesized, also known as when the chromosomes duplicate. And during the G2 phase, the cell synthesizes proteins and continues to increase in size. Thus, a cell grows during the G1 phase, continues to grow as it copies its chromosomes in the S phase, grows more as it completes preparation for the cell division in G2 phase, and divides during the M phase. Mitosis is the division of genetic material in the nucleus. It is an important process in the normal organismal development. Mitosis is broken down into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. During interphase, a nuclear envelope encloses the nucleus. The nucleus contains 1 or more nuclei, and 2 centrosomes have formed by duplication of a single centrosome. Chromosomes duplicated during the S phase cannot be seen individually because they have not yet condensed. Each duplicated chromosome appears as 2 identical sister chromatids joined at their centromeres, and the mitotic spindle begins to form. The mitotic spindle is composed of centrosomes and the microtubules that extend from them. The radial arrays of shorter microtubules that extend from the centrosome are called asters. The centrosomes move away from each other, propelled partly by the lengthening microtubules between them. At the beginning of prometaphase, the nuclear envelope fragments, and the chromosomes have become even more condensed. Each of the 2 chromatids of each chromosome now have a kinetochore, a specialized protein structure at the centromere. During metaphase, the centrosomes are now at opposite poles of the cell. The chromosomes convene at the metaphase plate. Anaphase, which is the shortest stage of mitosis, begins when the cohesion proteins are cleaved. This allows the 2 sister chromatids of each pair to part suddenly, so each chromatid thus becomes a full-fledged chromosome. By the end of anaphase, the 2 ends of the cell have equivalent and complete collections of chromosomes. During telophase, 2 daughter nuclei form in the cell and the nucleoli reappear. The division of 1 nucleus into 2 genetically identical nuclei is now complete. Cytokinesis, which is a process in which the cytoplasm of a single eukaryotic cell is divided to form 2 daughter cells, is usually well underway by late telophase, so the 2 daughter cells appear shortly after the end of mitosis. Meiosis is a modified type of cell division in sexually reproducing organisms consisting of 2 rounds of cell division but only 1 round of DNA replication. During prophase 1, duplicated homologous chromosomes pair and exchange segments. During metaphase 1, chromosomes line up by homologous pairs along the metaphase plate. During anaphase 1, each pair of homologous chromosomes separates and moves toward opposite poles. During telophase 1 and cytokinesis, 2 haploid cells form. Each chromosome still consists of 2 sister chromatids. In meiosis 2, the separation of the sister chromatids takes place, and 4 haploid daughter cells result, containing unduplicated chromosomes. After the 2 haploid cells have formed and separated, prophase 2 occurs. A spindle apparatus forms, and in late prophase 2, the chromosomes move toward the metaphase 2 plate. During metaphase 2, the chromosomes are positioned at the metaphase plate as in mitosis, and because of crossing over in meiosis 1, the 2 sister chromatids of each chromosome are not genetically identical. During anaphase 2, the breakdown of proteins holding the sister chromatids together at the centromere allows the chromatids to separate. The chromatids move toward opposite poles as individual chromosomes. And finally, during telophase 2 and cytokinesis, nuclei form, the chromosomes begin decondensing, and cytokinesis occurs. The meiotic division of 1 parent cell produces 4 daughter cells, each with a haploid set of unduplicated chromosomes. Comparing and contrasting meiosis versus mitosis. The DNA replication in mitosis occurs during interphase before mitosis begins, and in meiosis it occurs during interphase before meiosis 1 begins. The number of divisions. Mitosis only has 1 division, and meiosis has 2. The number of daughter cells and genetic composition. In mitosis, there are 2, each diploid, 2N, and genetically identical to the parent cell. And in meiosis, there are 4, each haploid, N, containing half as many chromosomes as the parent cell, genetically different from the parent cell and from each